Hello dear students. In this video, we're going to speak about tissue engineering. Let's start. What is tissue engineering? Tissue engineering is a part of the field of bioengineering. Bioengineering is a broad discipline which combines principles from both biology and engineering. It is sometimes described as taking an engineering approach to the study of biology. Within healthcare, bioengineers contribute in areas such as device design, physical therapy, and drug delivery. Tissue engineering involves investigating the biological, physical, and chemical forces involved in tissue development, injury, and wound healing. The goal of tissue engineering is to restore, maintain, improve, or replace biological tissues. This requires understanding what the healthy condition looks like and how to return damaged tissue to their state with treatment. To study this, tissue engineers must grow cells in a laboratory which behave like healthy cells grown in the body. Tissue samples grown outside the body using tissue engineering techniques are often referred to as tissue-engineered constructs. What is the difference between tissue engineering and regenerative medicine? The terms tissue engineering and regenerative medicine are often used interchangeably. Both focus on the repair, maintenance, and restoration of biological tissues. The key difference is that tissue engineering focuses on growing tissues outside the body. Regenerative medicine specifically focuses on how tissue engineering techniques can be used in a healthcare setting to repair tissue within the body. A lot of research in tissue engineering has the long-term goal of developing a construct that can be used in the clinic. Tissue engineering research is a necessary first step in regenerative medicine therapies. What are the principles of tissue engineering? The three defining elements of tissue engineering are the use of stem cells, a biocompatible three-dimensional scaffold, bioactive molecules. Stem cells are cells which are capable of developing into more than one cell type. The best known examples are embryonic stem cells, which can turn into any type of cell in the body. Stem cells in adults can differentiate into several different cell types, depending where they originate within the body. For example, adipose-derived stem cells, which are found in fat tissue, can differentiate into bone, cartilage, fat, and several other tissue types. The path a cell takes depends on many factors, including mechanical forces or exposure to chemicals. Scaffolds are three-dimensional structures which support the growth of stem cells into the desired cell or tissue type. Cells in the lab are often grown on flat surfaces or suspended within a liquid. A 3D scaffold is a closer match to the 3D environment of the body. It is important that the material used in a scaffold is biocompatible, that it will not damage the tissue it comes into contact with. This means the material must not be toxic, but also that it must not break down over time into small parts which might irritate the tissue. Materials used in tissue engineering scaffolds include collagen or certain protein chains. Scaffolds for tissue engineering must also be porous. Stem cells can only be coated onto the outside of the scaffold, so it is important that the scaffold is porous enough for the cells to move inwards towards the core as they grow. If the construct has cells only on the outside and none at the core, it will not behave like normal tissue. Bioactive molecules are substances which have an effect on living tissue. In tissue engineering, this may mean signaling molecules or growth factors which can influence how a stem cell differentiates. These bioactive molecules may be in the nutrient mix used to grow the cell in a lab. They may also be incorporated into the 3D scaffold during the manufacturing stage. Researchers are also investigating how bioactive molecules might be used to make tissue-engineered implants more effective. For example, a scaffold could release a drug which reduces inflammation or which helps the implanted cells embed at the implant site. How can tissue engineering be used in research in developing gene and cell therapies? Tissue engineering techniques are used to grow models of tissue in the lab. These have many uses in research. Studying normal tissue development. Engineering tissue can allow researchers to see how certain tissue types develop from stem cells. By controlling the environment, they can observe how certain changes affect the developing tissue. This allows them to answer very specific questions. When conducting a study in animals or humans, there will always be unknown factors which can't be controlled. In a tissue-engineered model, researchers can control changes to the environment and can measure the results precisely. Studying diseases at the tissue level. Scientists can tailor the lab environment to resemble a particular condition. For example, to understand how arthritis affects cartilage development and repair, scientists may add inflammatory molecules to the nutrient mix. Another way to study a condition at the tissue level is to collect cells from patients with the condition and compare the development of their tissue with cells from healthy patients. Testing therapies. Tissue-engineered constructs can be used to test drugs to confirm that they are safe for a particular tissue type or to see what effect they have on a disease model. That is the end of our video session. Thank you for watching.